Hi everyone, Devin here. We're going to be moving on to the third problem about mapping three linked genes for last week's recitation. This will be starting on the third page and moving on to the fourth page. Before we do that, just want to visualize something that would hopefully help you make some sense about what we're going to be processing in the subsequent slides here. So, three linked genes, and although this question deals with body color, nose color, and antler shape, I want you to just ignore that for a second and imagine we're just going to call these three other genes random three genes A, B, and C. You can imagine what we're dealing with are individuals again in which we have a heterozygous parent and a homozygous recessive parent. And what we're trying to do is see what happens as a result of the offspring. So what would happen in this scenario? Well, obviously you get to give one of the other from parent one, or you get to give one or the other of parent two, and that's what goes on to the progeny. So when we talk about no crossing over, what would be the case? Well, you could either give this to your offspring, or you could give this to your offspring. And so we could see, a combination like this, and then the other parent, obviously, maybe I'll draw this in blue in this case. The other parent can contribute this, and obviously the parent can only contribute, the second parent can only contribute that genotype, whereas this parent can contribute either of those. So when we look at what's the result in the offspring, it doesn't really make much sense to describe these things in terms of their full genotype. So you may have filled in something like this earlier, or something like this. And that's fine, but for all intents and purposes, if we're trying to think about what kind of variation there is, when we're always defining these linked mappage, linkage map problems, we always have a homozygous recessive parent. Really, all you have to worry about is what's the contribution of the heterozygous parent. So we're really only going to need to bother writing these things in terms of this or this contribution from the heterozygous parent. You don't really have to worry about the homozygous parent. So because of that, you don't only have to write out the full genotype. You can just write out, if there was no crossing over, what we would see is this or this in the resulting offspring. So that's one thing to keep in mind, is that when we're writing out what are the resulting genotypes, even in the progeny, we're really only saying with respect to what happened from the heterozygous parent. So, and we saw in no crossing over, it looks something like this. But now we can imagine a scenario where, I'll do a little better job of using some color here, we have some again, same parents, same contribution, and the initial parent, I don't really worry too much about the homozygous recessive parent anymore. Because again, any crossing over event that happens, whether it's a single or a double cross, you're always going to get the same resulting chromosome. But some unique things can happen. So let's imagine in this case we have a single crossover event occurring at this position. If this was the case, then we could see that the resulting genotype would be just follow the red line. So you would start at this position, you'd move down to this position, and you'd stay along that chromosome. So that would be one of the contributions. And so this would look something like an uppercase, lowercase, and a lowercase. And likewise, we could have the other chromosome mapped out to a lowercase, uppercase, and an uppercase. So we could see something like that. But that's just at one position. We could obviously have a single crossover event occur at the other position. So draw this out one more time. If instead we have a crossing over event occurring between B and C, well, now we have something slightly different in terms of the output. Sure enough, we have A, B as uppercase and lowercase c, as well as the other two lowercase followed by an uppercase c. So something like this. Those are the four possible contributions that the heterozygous parent could make depending on which of the positions in which the crossover event happens, whether it's between A and B or whether it's between B and C. But hopefully as a setup you can see that that's really the possible outcomes for the single crossovers. There's obviously one more scenario that can happen, and that's the double crossover. So you can imagine here again, same parents, where we have a crossing over event between both positions And if the crossing over event happens between A and B as well as B and C, well, now it's a little bit trickier to follow the line, but you come down here, and you have another crossing over event, so you come back up here. So this is one of the possible outcomes. So you could have, you could have this genotype, or you could do it the other way. I'll switch colors here just to make it clear. You could come up here, come back down, and in that scenario, we would have an alternative genotype. So if you look at all of those possible outcomes, there are 8 different genotypes that only the heterozygous parent is contributing to. And in each of these scenarios, it just depends whether a single crossover, double crossover, or no crossover event has happened within the heterozygous parents, gametes, if you want to think of it that way. So that's the setup, because the following scenario, when we try to fill in these charts, is really geared to knowing that we're talking only about the heterozygous parent, that we're not putting any information relative to the homozygous recessive parent. It's all about the heterozygous parent. To fill in each of these things, you don't have to fill them in as diploid genotypes, but simply haploid genotypes. So to remind you, we're talking about 3 different phenotypes in which we have Ds, which is representing body color. In this case, this was tan, this was dark. We had Rs representing nose color, so this was black, and a lowercase was red. And we also had now we've introduced our 3rd gene of antler shape, and uppercase meant flat, I believe, and the lowercase represented arched. So we see each of these kinds of phenotypes, and your job is really straightforward. If you see tan, write in the associated allele for tan. So in this case, we know that tan is represented by the dominant allele, so I'll just fill in tan in each of those scenarios. The lowercase allele for dark, fill that in, and you'd fill the chart in as appropriate for each of these. So what we're trying to represent here is not the diploid genotype of the offspring, but simply these are the 8 combinations that would result from a heterozygous parent's contribution, whether there was 0 crossing over events happening, a single crossing over event happening, or a double crossover event happening. So you'll see there's 8 boxes here. If you look back to the previous slides we were just talking about, you'll see there's also just 8 combinations that could happen. And the key thing is to sort of look at the numbers. You really want to focus your attention on the number of offspring, because this is what's sort of a dead giveaway when you're trying to figure out which of these combinations of D's, R's, and A's, whether the uppercase D, uppercase R, uppercase A, or lowercase d, uppercase R, uppercase A. Which of these things is the parental contribution? And it just comes down to the number of offspring. We've said this in an earlier YouTube clip, and we've said this in class multiple times. The dead giveaway is the relative number of offspring of a given genotype. And the ones that are the largest numbers, and obviously 450, 460, much, much, much larger than any other kind of combination of D's, R's, and A's, these are the parental classes. And as we've mentioned before, it's the parental classes relative to the heterozygous parent. So these are not only the most abundant because they're the ones that require no recombination, but they're also telling you another piece of information, which is these are the parental genotypes of the heterozygous parent. So if you were going to try to think about what that would look like, we'll draw that out in a second, but actually just do it now. The parental genotype must have then been something like this, where it's D, R, and A. That's this example, as well as they're obviously all D, R, and A. Lowercase d, lowercase r, uppercase A. And that's this one. So we know that for the reason being that there's a very low incidence of recombination, or at least it is to say that recombination will never be greater than the relative contribution of no recombination. So these must be the parental genotypes because they're the most abundant. We can write them out as individual chromosomes, and hopefully what strikes you as this must be true is that if we compare each of the D's, each of the R's, each of the A's, there's one dominant and one recessive allele. So we said this was the heterozygous parent. Sure enough, that looks like a heterozygous parent. So what do you do with the rest of the information? Then it sort of becomes a little trickier. Which one of these represents single crossover events? Which one of these represents double crossover events? I think the other extreme is the easier case to sort of explain. Double crossovers are far more rare than single crossovers, and obviously much less rare than no crossing over. So these represent the double crossover events, since the 0 and 1 number of offspring at each of those genotypes of dRNA are far, far less frequent than any other. And that just leaves us with 4 other classes of single crossover events as the sort of intermediate classes of abundance. So this is one way to think about the information. You can convince yourself that these are also single crossover events if you go back and draw out what these parental chromosomes would look like, but there's a big but. We don't know the order of the D, R, and the A from the outset of the problem. Although we've drawn D as the first allele, that's not necessarily the case. And actually, in this case, we're trying to figure out what is the order. Does D go in the middle? Does R go in the middle? Does A go in the middle? And we've drawn it here, R is in the middle, but that's not necessarily true. So how do you figure it out? We can figure it a couple of different ways. And one of the ways is to sort of visualize what would happen. Again, given that we know that there are certain frequencies of combinations of alleles, certain combinations of these dRNA genes, we can test to see, if R was in the middle, what would happen? If D was in the middle, what would happen? So let's just take a minute and test that. So okay, so in this case, we're going to imagine that we had it right all along, and I'm going to quickly just remind you that the parental genotypes here, D, R, and A, as well as D, always R and A. If there was a crossing over event, and it's a single crossover event here, and this was the order, if we said that R is the thing in the middle, then what we would get at the outcome of that crossing over event was something like this, or something like this. We would get these in some relatively high number. If this was initially our parental genotypes, and to remind you, I'm going to jump back a slide, just to remind you that these were the heterozygous parental forms that we would have found from the chart. This was the one that had 451, this was the one that had 466 offspring. If this was to happen, if this was the order, DRA was the order of the genes on the chromosome, then we would see that if there was crossing over as a single crossover event, we could have these new combinations in relatively high proportions. But if you look back to the chart, you'll find that that combination of an uppercase D, a lowercase r, and an uppercase A, uppercase D, lowercase, I have to sort of show you the slide here. That has a total number of offspring of 0. And likewise, if we look back to the original chart of the lowercase d, uppercase r, lowercase a, that has a number of offspring that's 1. And that doesn't make any sense because we wouldn't expect a single crossover event to have the lowest abundance. So R probably isn't the thing in the middle. So we could try keeping the exact same parental genotypes, but we're just going to reorder them. So I'm going to shift the order in which these things are on the chromosome. So instead of R being in the middle, we'll put A in the middle. We'll keep the letters the same, but we'll just simply rearrange their positions. So I still have an uppercase D here, I still have a lowercase a, and I've just shifted it to the middle, and an uppercase R, and I put it to the end. Likewise, we'll do the same thing for the other chromosome. We'll keep the D's together, but now we've obviously moved the positions of each of these. So again, imagine there's a crossing over event, and the single crossover event happens here. And we haven't shifted anything about the actual genotypes of the heterozygous parent. All we've shifted are the order in which those genes exist on a chromosome. So what we would get at the end of this is an uppercase T, an uppercase A, and a lowercase r. And if a single crossing over event happened again at that position, we would get a lowercase t, a lowercase a, and an uppercase r. Now you can ask yourself, did anything change? And, well, we've sort of rearranged the letters, but despite rearranging the letters, the outcome in terms of what those things represent, who does that look like? Well, big D, big A, little r, that's this. We say this is still the same combination of alleles. And sure enough, this combination is the same as this one up here. So just by reordering the positions of the letters, we don't actually change the outcome in terms of what this chromosome and its combination of genes represents. So this can't be the case. We can't have an A or an R in the middle. That can't be the right scenario. So what if? What if we have the same heterozygous parent again, and we don't position the A or the R in the middle, but in this case we position the D in the middle? And what can happen? Well, let's imagine there's a single crossing over event happening here. The resulting offspring can follow this pattern. We would see that there's a uppercase R, a lowercase d, and uppercase A, and in this case we'd have a lowercase r, an uppercase d, and a lowercase a. So what does this look like? Well, let's just put it back in terms of d's and first, since that's how we drew them in the table. We'd have lowercase d, an uppercase r, an uppercase a, in this case an uppercase d, a lowercase r, a lowercase a. Now, looking back at that table, what we find, sure enough, is that the combination that we're representing here, big D, little r, little a, there's 17 offspring of that order, and there's 18 in this order, looking back again at the initial table on page 3. And that's kind of the middle ground. That's not the least frequent offspring. That's not 0 or 1 like the double crossover. This certainly looks a lot like a single crossover event. So it must be that if we position D in the middle, we start to get the kinds of combinations we would expect. So this is one way that you can convince yourself that D is certainly the answer for question F about which one's in the middle. And another way that you can do it, it's a little bit different, but what you can do is look back to the table and write out what you see for the double crossover events and the parental events, parental classes, I'm sorry. And so if you see something like this, where you have tan, that's your D allele, red, that's your lowercase r, and flat, that's your uppercase A, we'll find that the number of offspring is 0, and this represents one double crossover. Compare that to a parent, and it doesn't really matter which one you do, but compare that to a parent. Let's say that the first parent looks like this. It's black, so it looks like that, and it is arched, so it looks like that. And we have 451 of those. So one of the other ways you can figure out, if you don't want to try to redraw each of these crossing over with chromosome kind of visualizations that I just went through, is compare a double crossover with a parental, and then look at the combinations of each of the associated phenotypes or the individual alleles. In this case, there's a difference between the double crossover and the parental. At the A allele, there's a difference at the R allele, but there is no difference at the D allele. That's another way of tipping off that the D is the position that should be in the middle. You can do the same thing for the other pairs. You'll find that nothing is changing for the D's in the other combination. And really the lesson here is just which one isn't like the other. In this case, this is the one that's not like the other two. It's not changing, while these two are changing. So that tells us that positionally the D gene must be between the R and the A flanking on the outside. So if you want to go through each of the problems, then we've been able to fill in part A by filling in that table. We've been able to fill in part B for filling in the parental classes. Those are simply the ones that are the most abundant. That wasn't really about identifying anything special about a genotype, just simply saying the parent class is going to be the one that has the most abundant offspring. We filled out the single crossover events as the intermediates, and the double crossover is the least abundant of all. So to directly answer part C of that, when you're trying to figure out what the alleles were on each chromosome of the heterozygous parent, that's simply just go across the table, and if you find that the parental class that you filled out that had 451 in it, you'll find that the first parent is this combination excuse me, and you'll find that the second parent is obviously heterozygous, so we'll find that it looks something like that. That would be the answer for part C. We can imagine that now, knowing that D is in the middle, we can sort of draw these as we've been drawing them all along. We could see it looks something like this. That would be our chromosomal diagrams for the heterozygous parent. We identified the double crossover events, and we can figure out what those chromosomes would look like. And then we were able to determine that D was certainly in the middle. That was the answer for part F. And what you're asked to do at the end of page 3 is to rearrange all of the columns so that we have the right gene in the middle, and what you'll see is that we've now positioned the D gene in between the R and the A. You might be asking yourself if R or A needs to go first or last. It makes no difference for the context of this problem. If you're really trying to think about where R and A are positioned in the rest of the chromosome, what you need to do is grab even more genes and perform more crosses. And that could tell you something about where A and R are in the context of the entire rest of the physical unit that is the chromosome. The other thing you'll notice is that we can identify that there are single crossing over events between R and D and A. And again, if we've got D in the middle now, and you think about what your parent looked like a minute ago, it must be that if there's going to be crossing over going on, that a single crossing over event here must be between R and D, so that would be something like that. Or you could have a single crossing over event between D and A, something like that. You could not have a single crossing over event between A and R without incorporating something to do with D. And hopefully at this point that's kind of obvious why you couldn't just draw that out. Okay. So on to the fourth question, or the fourth sheet, and we're trying to calculate some map distances out of this information. So what you need to do is still pay attention back to the previous table. What you would have found are the relative number of offspring produced among the entire set of progeny. So if you're going to try to map out the distance from the middle gene to each flanking gene, since D is in the middle and we've got R on the outside, and what we're trying to figure out is what's the distance between R and D, what's the distance between D and A. How you do it, let's just say for this distance first, what you do is you add up all of the single crossing over events that are relative to R and D, and so that would be 18, 17, as well as the double crossover events, because those also involve R and D. We'd add those all up, and we would add it relative to the total number of offspring that are produced for all possible combinations. So you'd take the 8 individual offspring classes and add them all up, and you'd find that adds up to 1,000. Take that number, multiply it by 100 so you have a relative percent, and you'll find this turns out to be 3.6% or 3.6 map units. And that's the way of calculating the distance between R and D. Between D and A works just the same. The only difference is that you're going to add 2 slightly different numbers. You don't have the single crossover events with respect to R and D, but you want the single crossover events with respect to D and A. So in this case, you substitute 18 and 17 for 22 and 23. If you look back to the table, those will be the 2 single crossover events that are associated with D and A. But again, you include the double crossover events. Why? Because those are also associated with crossing over between D and A. Multiply those up by 100, formula looks just the same, and what we end up with is 4.5 map units. So that's great. That tells us something about the frequency with which we would expect crossing over or combination to occur between R and D, whether it's with a single or double crossover event, and what we get out of this is the relative distance between those. You should note that the 3.6 map units looks a whole lot like what we calculated much earlier in an earlier clip, in which we were just talking about the distance between R and D genes, so it does fit. The next thing you want to do, this is part H on the worksheet, is to calculate, to draw out a genetic map based on these calculations. We already know the positions now of R relative to D and D relative to A, so we can just simply draw on our map. Here's R as a gene, it's 3.6 units away from D, which is 4.5 units away from A. And that would be the way you can draw a genetic map using that data. The last thing to do for part I and part H are to calculate the expected number of deer that would have a double crossover event occur. And you can use the product rule here. So what we're trying to figure out really is how many double crossovers are there that we would expect. And you can do that just by taking the product of each of the distances between the R, D, and A alleles. And if you know that the distance at this point is 3.6 map units and the distance here is 4.5 map units, you'd expect that the number of times you get a double crossover is the product of each of those. And so how we actually calculate that and plug this into a calculator is 3.6 as a percent is 0.036, 4.5 as a percent is 0.045, and the product of each of those turns out to be an even smaller number, or 0.16%. That is to say, what we would expect is the number of times we get a double crossover is 0.16%, or you can think of it in terms of a slightly more of a whole number, 1.6 offspring in 1,000. Since we had a total number of offspring that was in 1,000, we can put it in the same context. So not very many. And what that leaves us with, that's the sort of way you would work through part I. Again, double crossovers being very rare. What you want to figure out in this last bit is something about that's what we would expect, but we know what we saw, and we can sort of look and say something about the difference between what we observed and what we expected. And the way that goes down is you can calculate two things that are basically reciprocals of each other. The first thing you can calculate, and this is how we go through the last part on page 4 and part H, is you're calculating something called the coefficient of confidence. And it's really just, it's a ratio. It's just the ratio of the observed number of double recombinants relative to the expected number of double recombinants. So we've calculated the expected, that was right here, and we have an observed number of double crossovers. So looking back, we can calculate this thing called the coefficient of confidence, and it's going to just be observed double crossovers over expected double crossovers. So we have here our expected, so the question is, what's our observed? Well, look back to the data table, and you'll find two different classes of double crossovers. One was 0 and one was 1. So you add those together, and 0 plus 1 will turn out to be 1. So let me put this on a new sheet so this makes a little more sense. We have observed over expected double crossovers. We have 0 and 1 double crossovers in our real data, and we have an expected 1.6 out of our real data. So we get 1 over 1.6, and finally that turns out to be 0.625. So this is a number that's less than 1. It doesn't always, it turned out, have to be less than 1, but by and large you're going to find that the coefficient of coincidence, is generally less than 1. It's really, really rare if it's greater than 1, and if you do that, we've been very mean on a test to you. So less than 1, what that means is that something is less than what we expect. There are fewer double crossover events occurring than what we would expect, and there can be reasons for why that is. Probably the simplest one is simply to say that if we see fewer observed double crossovers than we would expect, then when we think about these genes as physical units, that when we had that heterozygous parent all along, that if there is a double crossover event here, a single crossover event, that it's influencing whether or not another crossover event can occur at that position. And what we're seeing is that we have fewer instances than we would expect. Maybe it means that when there's a crossing over event here, it reduces the likelihood that there is crossing over occurring here. That could be physically because it's harder for the recombination apparatus to get in and bend the molecule. Maybe they're very close. There can be other reasons, but for now, just know that when you have a coefficient of coincidence that's less than 1, it just simply means that there's something going on that's inhibiting the or reducing the probability of a crossover occurring near this neighboring region, the DNA region. But there's a term for that, and that's where the last point here is that thing, when a single crossing over event occurs, lowering the probability of a neighboring crossing over that is a second crossing over, that is, by definition, interference. And that's why we think about these things, these numbers, as reciprocals. If you want to know what the interference value is, it's 1 minus the it's one minus the coefficient of coincidence. So the interference likelihood is 1 minus the value we just calculated. And we find that it turns out that you can calculate it as a number directly, and it really it's a relative number that tells you something about, again, the likelihood that the probability of a crossing over in an adjacent region is being reduced by virtue of the fact that you have one crossing over event occurring in the first place. So more interference, more likely that you reduce the double crossover outcome. Less interference, less likely that you have a reduction in double crossovers. And again, with respect to 3 different genes. If you had genes that are very, very, very far apart, you might imagine that interference starts to drop simply because that sort of physical getting in the wayness of very close neighboring crossing over events just isn't happening. So that should get you through the rest of the worksheet. If you have any questions, feel free to post them on Slack. Ask me, ask Andrew, ask Jen, ask away. But hopefully this will get you through all the tough stuff that we presented in recitation. Have a great week.